Butter is a saturated fat. It's solid at room temperature. It's coming from animal fat.
Is the phospholipid bilayer, are they saturated or not saturated?
The fatty acid tails?
Yeah.
I don't know. They're different. It's a different way to think about it, I think. So, I don't know. What else? What's the function? What's the ratio between hydrogen and
Or
oxygen?
energy, right?
What's the ratio between hydrogen and oxygens in these? Way greater than 2 to 1. There'd be lots of hydrogens. These are all like hydrogens. And then there'd only be like a couple oxygens. All right? That's one easy way. Oh, it's even in that picture. That's one way to tell the difference between a carbohydrate and a lipid. One, these are very linear. And two, there's lots more hydrogens than oxygens, greater than 2 to 1. I'd write all this down if I were you. I'd be making an awesome study guide. That's what I would do if I was you. But if I'd be you, then you'd be me. And then what?
We wouldn't have a final.
Did you already write the
There's no final?
The function. Maybe long-term energy storage. What else?
Keeps you warm.
Insulation for your body or also the organs. The layer around your organs to protect. What? Yeah, waxes. What else were some other weird examples of? Vitamins were an example. Steroids were an example. Oils. What did you say? The leaf. Oh, the cuticle, the waxy surface on the leaves. On leaves are a lipid. It's helping protect it.
And isn't it also for water retention?
Yes. Because you're going to make it harder for water to evaporate or leave from that cell. Because it has that waxy surface. Anything else with lipids?
They're hydrophobic.
Okay, great. Hydrophobic, right? Our test for the lipid, right, was to mix it with water and it rises to the top. Hydrophobic does not mix with water.
It's a fire retardant.
Say it again?
It's a fire retardant.
Okay. What was... There's one more question on this thing. It has something to do with... What is glycogen?
Sugar, protein.
A starch that is used in storage in plants, or no, glycogen is used in storage in animals.
In animals. So that was a carbohydrate?
Yeah.
All right, so that's one that I forgot. Sorry, it just popped in my head. So glycogen was used for storage and carbohydrates in animals. What was in animals. What's used in plants?
Starch.
Starches and cellulose. What's the other one?
Cellulose is used for the starch.
Right, but it's still that polysaccharide in starch in plants. So you should know those. All right, sorry. I think we're good with lipids. Proteins. What's the function of a protein? For energy, right? I go to the gym and I drink a bunch of protein shakes and then I have so much energy I'm just about to...
Muscle function.
It's not to get energy though, right?
It's muscle function.
The protein shake is going to have a lot of carbohydrates in it and a lot of fat in it, and that's going to actually give you what the energy you need to do those things. So the muscle repair, any structure and like any functional thing that you're trying to do, it's going to help build muscles, repair things. What else? What else? That's it? Okay. What's the structure of a protein look like?
Oh, there's many, aren't there?
There's like primary, secondary,
Lots of
tertiary.
structure.
So the primary structure, we just talked about this the other day.
Precise sequence of the amino acids.
The sequences of the amino acids, so how those amino acids, in what order they are, determines that primary structure.
And that like determines what that
Yep, and then what else determines the function? The shape, so how those things are shaped, right? There was pleated, which were the folds, there were helixes, which are the spirals, right? How all those shapes are determines the function of the protein. What does it look like? What does the structure look like?
Yeah.
What's the structure of a protein? There's a central carbon, an amino group, a hydroxyl group, hydrogen, and then an R group, right? Those side chains are what determine the different functions of all the proteins. All right, so they were always looking like, there's always a C here, and then some sort of N something, some sort of H something, some sort of H, O something. And then this R group, right, is what actually determines, and this could be really long.
Can you repeat the muscle protein functions?
I cannot, but maybe your classmates could.
To build muscle
Good.
It's structural purposes.
And for all the functions, right? Proteins are doing all those things. What were letting things in and out of the membrane? Proteins. They're the things that do. What are the monomers? Amino acids.
Is it like the R group and everything? Does that change the function of?
Yeah, because that's going to change the whole shape and what it is able to do. Nucleic acids. What are the functions of nucleic acids?
Store information.
Store information, transfer information. What kind of information?
Everything.
Genetic information, how to build proteins.
Yeah.
So you should write for those. What's the structure of DNA?
It's a
Double helix.
That double helix. RNA, single-stranded. Monomers of nucleic acids.
nitrogen bases.
Nucleotides. We did not. So that's the monomers, and they're made up of the nitrogen bases, phosphate group, and the sugar deoxyribose or ribose, which are pentose sugars. Right, they have that 5 side. What else with the chemistry? Hydrogen bonds?
I need to know the specific heat and stuff like
No.
Where?
heat
Isn't
of vaporization.
Nah.
Aren't like van der Waals forces somewhere in DNA?
Yeah, Van der Waals forces are important. Those are super, super weak.
Where are they
Bonds.
located?
So it's a really weak attraction that keeps like DNA kind of closely together
Okay.
when it's stacked up on each other.
Don't we-
Covalent bonds were really strong bonds. So whenever you see covalent bonds, that means it's like permanent, pretty much. Denaturing what gives the protein its shape, right? Or what gives it its function? It's that shape. So if we denature a protein, that means that we've
Yeah.
changed some sort of environmental condition, right? Like we've changed the temperature, changed the pH, and that can denature it. It loses the structure.
Can you optimize the structure?
No, because it's already optimized for
Or
whatever
what if it's not for some reason?
it is. I don't know. Maybe.
Wait, what
I don't
is
know.
a pH?
Probably
again?
not. But then if we change that, if we denature the protein, and we lose that quaternary or tertiary or secondary structure, maybe the primary structure is still there. So if you return to the environmental conditions, that primary structure might still be intact, unless you like really heat it up or something, cook your egg all the way, and then it denatures even that primary structure, and then it's done.
The if you change the pH.
That's what I was talking about. If that primary structure is still intact,
So what are the reversible ones?
that's that example. You could denature it, but only denature like the how it's. As long as those amino acids are still intact, it should still be pretty good.
The pH.
pH is measuring kind of like acidity, right?
Yeah,
The
like
measurement of
acid.
free hydrogen ions. So low pH is acidic, high pH is alkaline,
Like a base.
like a base. It's all about the base. Cell structure, function, and membranes and transport.
Can you remember it was like the something like potassium channel?
Whoa. Yeah.
What was that?
Sodium potassium pump.
Yeah, that was it.
So that was, there's passive transport, right? And there's active transport. And I said, all we really need to know for active is that sodium potassium pump. We're gonna talk about that later.
Yeah, it requires.
That's all I want you to know right now, is that's the example of active.
When later?
Next semester.
Oh, okay. I thought you were before the final.
No.
Like, when-
Sodium potassium pump is using energy, right? So it's going against the gradient, right? It had to like pump that hydrogens across the gradient, maybe. All right. What else? What does the gradient mean?
That change it.
The concentration.
Concentration gradient? What's that?
The water shifts to balance out the.
It's a difference in the concentration between the, what's it called, the end of the cell membrane. So like on the outside of the cell membrane border could be a really low or really high concentration of hydrogen, and then be the reverse of that on the other side of the membrane.
So the more hydrogens there are over there, the bigger that gradient gets, the more potential energy there is.
And it has like a natural tendency.
All right, so when we get down to respiration and photosynthesis, it uses that chemiosmosis, right? Uses that principle and then shoots all the hydrogens through that ATP synthase, and that potential energy of that stuff rushing through creates more ATP. In the membrane ones, right, the more salt that was outside of the cell, the bigger that concentration of salt is outside of the cell, the bigger the concentration gradient. Now water is going to move out even faster because there's more potential energy.
Can you go over the cell structure function?
Cell structure?
Yeah.
So what do you want to know?
Everything?
I don't even know what that means.
Oh, that's right, yeah.
What?
You know how we did that worksheet where we like colored in the organelles?
Yep.
Can you put that?
You want an organelle worksheet? Yeah, I'll put one up there. You can find a million of them online too.
Okay.
So what are the organelles that we need to know?
Ribosomes.
Ribosomes, those are pretty important. Centriole,
Mitochondria.
Golgi apparatus.
Lysosome.
Rough
central vacuole,
Smoothie R.
rough ER, smoothie R, what does smoothie R make?
Smoothie R is there?
No ribosomes on.
Yeah.
So what do they make? Ribosomes make proteins, right? So the rough ER is making proteins, smoothie R is making,
Fat.
Not protein.
what's another one that they can make then?
Fat.
Lipids are what they make.
Is it lipids or fatty acids?
One.
Both would go under that. These fatty acids will build different lipids. What else? Nucleus, prokaryotes versus eukaryotes, plants versus animals.
Prokaryotes don't.
Eukaryote.
Prokaryotes don't have nucleus, they have nucleoids.
Or like bacteria cells and
Bacteria cells,
animal and plant
Are
cells.
prokaryotes only animal
single
cells?
celled usually.
Are prokaryotes only animal cells?
Prokaryotes are bacteria. Eukaryotes are plants, animals, highly developed things. Yes.
Like, can't prokaryote also be like mold though?
Yeah, it's true. Not really. We'll talk about more about that. Get as far as we can.
If we don't, we're like in a session with Sunday or something.
Maybe a Monday.
On Monday.
Monday's easy.
What time on Monday you guys want?
6, 5, 5.
5, 4, 6, 3, 2, 1.
I think you'd want more comprehensive.
4 to 5. 4 to 5.
Yeah,
4
4
to
to
5.
5.
4 to 5.
Yeah, that's a good time.
What time is your last final out technically?
1. It ends at 3.
3:30?
Yeah, so I say 4.
So then you gotta digest a little or geek. So like 4? 4 o'clock?
4 to 5 is perfect.
Now I have Mr. Tennyson for his regular